electric, chemical, electric, electric, chemical, electric, electric, and so on, right? And this information that's being passed down through these is going to go in this direction because the information flow is always going to go from the dendrites, the cell body, toward the axon, right, toward the axon terminals. And neurotransmitters, this is only going one way, right? There's no backward signal like you might have in an electric signal because of this, right? So you get this one-way information flow. And so that is what's happening at the neurotransmitter, at the synapse right there. And this is what's happening in a couple of different neurons, right? And so we'll have the presynaptic neuron over here for this synapse, right? And the postsynaptic neuron. So for synapse 1, there's a presynaptic, here's your postsynaptic. But on the next synapse, over here at synapse 2, the next one down the line, this postsynaptic, which was the postsynaptic neuron, is going to be considered the presynaptic neuron because it's going to be the one that sends its signal to the next cell. And then this will be the postsynaptic one. Then it will go on and on like that. Now, this seems kind of silly. Why do you just keep on passing a signal from one to another and another, right? Gets complicated, but this is what is happening in your brain right now, all around. It's just bouncing around these signals, passing them from one to another. Cells trying to influence and communicate with the next cell or everybody that they're connected to. And somehow that results in consciousness. All right. So the last part of that is that if that chemical, the neurotransmitter, was left in here, it would keep on opening up these channels and you'd keep on having sodium or chlorine, whatever, into there. And you'd keep on having this cell activated, right? And you don't want that. You want to be very controlled. So once you've done with that action potential, you want to clear these neurotransmitters out. And this is an important part, right? Because this is a lot of how drugs and poisons and stuff work. I mentioned last time, Botox, for instance, prevented this release of neurotransmitter into the cleft right there, right? Other ones we'll see are going to maybe inhibit these enzymes, and then more neurotransmitter might be here. So there's a couple different ways that we're going to clear this neurotransmitter, right? Number one is going to be uptake, right? Either the own cell is going to bring it back in and recycle it. That's called reuptake, right? It's going to bring it back in. That'll clear it. And it's a nice way to recycle it. Or a glial cell, like an astrocyte or something like that, is nearby. You'll always see glial cells around the synapses, and they could take them up and clear them out and recycle them and maybe give them back to the neuron over here. So those are two ways uptake. Remember this reuptake later when we talk about serotonin, right? We'll talk about serotonin reuptake inhibitors and how they are used for antidepressants and anti-anxiety medicine. And then there is also just diffusion, right? You could have, enzymatic degradation, as we talked about with the acetylcholinesterase. It just breaks down it. And so those are no longer able to bind the receptors. So it breaks it down. Or it just diffuses out of the cleft, out into the interstitial fluid to be taken in eventually by the blood vessels, right? So those are ways you can clear the neurotransmitter from the synaptic cleft. Let's go back to last time, and we talked about those graded potentials, right? Here is your synapse over here. It was releasing neurotransmitters over here onto the receptors, opening up ion channels. That resulted, in this case, into a depolarization at this point. Right over here, somewhere around here, was the release of neurotransmitter, the flood of, in this case, sodium ions, for instance, inside the cell. And you went from a -70, and then your difference in between what was inside and outside started to change, right? It started to become depolarized, because that means it started going from -70 up towards 0, right? And if you're at 0, you're not polarized. Doesn't mean it reaches 0, it's just toward in that direction. That's a depolarization. And it's all based on the fact that signals went down, then all the sodium that was out here started to rush in over here, causing that positive influx. So you're looking at it over time. Here was the start. It took about half a millisecond or 5 milliseconds. We're maxed out at about -60 in this case, and then started coming back to your normal resting membrane potential because of leak channels and stuff like that. Sodium, potassium, ATP pumps, right? You just went back. This is just a little bump that kind of spread locally around the cell right here. And remember, it decreased as you got farther away from that stimuli. So that was what we looked at last time. And we saw that there could be a sub-threshold greater potential, that is the stimuli. Even if it was high enough, pretty high over here, -40, by the time it reached the so-called trigger zone, right, it was below the threshold. Remember, the threshold was whenever those voltage-gated sodium channels opened up that were all around the trigger zone. They were all over here, voltage-gated sodium channels, right, that we're going to open up, and then that would start the action potential. But to do that, it needs to be above here. And by the time this reached over here, it wasn't strong enough. So that was a sub-threshold graded potential. If you have a stronger stimulus, and we'll see what that means, right? It could be a very strong one, right? Maybe for whatever reason, it was very strong, and that was strong enough that that wave, even though it got smaller, it was still above so-called threshold. And then those sodium channels with their little activation gate, right, sensed that and opened up and allowed the sodium inside the cell right there. Because you had all around here, you had those voltage gated channels. Now, that would be a supra threshold. It doesn't matter if it was like this when it reached it, or it was like this, or just barely reached it. If it was above threshold or above, it was going to open up those channels, and then your action potential would start. So how do you get these supra threshold stimulus? There's a couple ways which we'll talk about here. And basically they all relate to summation, right? What happens at the trigger zone over here, right, depends on what's happening all throughout the cell right here, right, in order to reach this trigger zone. This is where it's all going to be summed. This is the integrating center. It's going to take everything here into consideration, whether it was a weak stimulus, strong stimulus, or, as we'll see, a couple of different types of stimuli, right? But a typical neuron in the central nervous system is actually receiving not just one, but thousands of input from other cells. Remember, there's like billions of neurons and trillions of synapses in the body right there. And so to integrate these inputs, right, that's going to be the result of all the postsynaptic potentials in the postsynaptic neuron all around the cell body right here. And the more excitatory signals, and here we're only talking about excitatory, the greater the chance that this threshold will be reached, right? Instead of this little thing or this little thing, you've reached it over here. So there's two types of these summation, right? You either could do it spatially, that is more than one neurons are firing excitatory, or temporal, that is one neuron is going to be very persistent and keep on sending you it, depolarizing, depolarizing, depolarizing until you actually reach threshold, right? So this temporal sensation is that one neuron that's just nagging you a lot, right? Just keeping on, are we there yet? Are we there yet? Are we there yet, right? It's going to do it over and over again, right? So you have a stimuli from this one neuron over here. It comes in and it gives a little bit of a excitation, but not enough for it to reach threshold. And then, because this takes a certain amount of time and it comes back down, it waits a little bit and then it hits it again. And then you get another little bump. And it really doesn't matter, right? They have not summed up together. That is, they haven't, both of them together, if they're spaced out enough, they're not going to cause any difference. However, just like our neuromuscular junction, we saw the same thing. If we get excess action potentials very briefly, you get a stronger contraction. So instead of, this between impulses, you have a very short distance. In this case, it hasn't got enough time to relax fully. You've depolarized. The next one hits, right? And when that next one hits, it reaches that threshold. Again, that's where your voltage gated sodium channels sense that voltage change and open up, and then they blast open, and then you get your action potential. Increase in the number from a single one, right? In a very brief period. That is temporal summation. That's one way you can get a neuron to fire, right? You got a lot of stimuli. This one's very excited, wants to really tell this guy, come on, come on, come on, come on, go, 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 right? And finally, it summates right there. On the other hand, you can also have a whole bunch of guys talking maybe sub-threshold, just a little bit. 3 or 4 guys talking quietly is kind of like one guy talking loudly, right? In this case, there's 3 different neurons coming in, a little depolarization here, a little depolarization, a little here, and then it adds up, right? Just like on the ocean, if you have a kind of bunch of different waves, they join up together and they get bigger and they kind of carry along there. All right, so summation of postsynaptic potential response to stimuli that occur at different locations all over at the same time, they're going to cause this threshold to be reached, right? So instead of this one guy going over and over and over again, they just do it very lightly, but the summation of these excitatory ones cause that trigger zone to be activated and the actual potential to fire. So that'll be spatial summation versus temporal. And you can do this also. Over here, you notice there's 3 different neurons in this one here, but this is one of the reasons of this branching off right here. Now you're getting, this was branched off even more, you'd have like 4 times the influence on this neuron right here. This is one way a single neuron could actually influence another cell more than just having a single synapse. This is all communication here, right? This is, go back to what we saw with that pathway with the sensory information going in. I'm going to go back to that picture over here. This is showing a single neuron in these places right here. But as I said, when you're sleeping right here, maybe you have, or you want to become more aware of stuff that's going on in your body. This doesn't have this single input. It may have a whole bunch of cells going into it. And it only fires when a whole bunch of ones are talking to it, or the same thing right over here, right? So you can get this kind of effect, as you're passing through, going up there and you're filtering information and you want to either maybe tone down the information coming in or pay attention to the information coming in, right? So that's temporal and spatial summation. And in this case, these are all ones that are exciting them, right? These are all opening up sodium channels, causing a depolarization in here, causing this excitatory. But when we talk about summation, what's really going on is that there's a combination of excitatory neurons and inhibitory neurons over here. So these, what they usually have to do is overcome the inhibitory neuron over here, or the inhibitory neuron has got to stop causing its inhibitory signal for these ones to go through. So remember I said most of the neurons in your body are interneurons in your brain and spinal cord. Most of your interneurons are actually inhibitory interneurons. For some weird reason, right? Most of your stuff is about inhibitory inputs to cells, to your other neurons right there. And it's about releasing, like stopping this signal right there so that these can go through, which is a lot of what your brain function is doing. So this brings us into this next topic right here. Excitatory postsynaptic potentials, EPSPs, and inhibitory postsynaptic potentials, IPSPs. Right, so for any individual neuron that's being communicated with, they got all the synapses all around their dendrites, their subbody, and they're being influenced by, remember, thousands, 100,000 neurons. There are excitatory neurons, excitatory input, and inhibitory input. And your The summation is going to be the sum of your excitatory and inhibitory, right? So for a action potential to happen, right, to reach threshold, the total excitatory effects are greater than the total inhibitory effects. And it was, not just the graded depolarizations, right, but enough so that you actually reach threshold to create that. A lot of times you just kind of make the cell partially depolarized in certain regions. You make it more excitable, more likely to fire when you're putting it in, but not quite enough to cause that action potential, right? That's just your normal EPSP, standard EPSP, excitatory postsynaptic potential. And then there's those inhibitory postsynaptic potential. And again, if the total input is more inhibitory than it is excitatory, then you go into this hyperpolarization state. That is, you go from -70 to say -90 instead of going upwards towards 0. And that would be a hyperpolarization, right? You're going down right there. What that does is make the cell less likely to fire when this guy tries to influence it. So that's your inhibitory postsynaptic potential, and that's going to cause a hyperpolarization of the postsynaptic membrane. So this is what we started talking about a little bit. I said put a pin in this for later. If the response makes the membrane less polarized, that is towards 0, it's depolarizing, graded potential. If it brings it down from -70 towards, say, -80, -90, it is a hyperpolarizing graded potential. Right? So mostly we were talking so far about these depolarization ones. Now we're going to kind of add this whole picture into it as far as this communication goes. So these, excitatory postsynaptics is going to be caused by the particular type of neurotransmitter and its receptor that is going to cause either a depolarization or a hyperpolarization. And so in the next video, we'll look at the ion channels in the postsynaptic receptor and the neurotransmitters released from the presynaptic neuron to understand how those hyperpolarizations and depolarizations will happen. All right, see you next time.